Okay, hello everyone. So welcome to my YouTube channel and to this lecture today. We have a lot of work and it's going to be a huge lecture because we have one very important chapter to go through and to discuss to make one step further from amino acids and to continue our story with polymers of amino acids, which are peptides and proteins. So we're going to discuss about the attachment of amino acids to each other. We're going to talk about the structure of proteins, about classification. So please get ready, and we have a huge work ahead of us. So let's start. So we know that actually proteins are the most common molecules in cells, and you know this fact from the introduction to biochemistry. So if we don't take into account the portion of the molecule of water, which you know it's like 70% of the body weight, the rest of the molecules, which are counted like a dry mass, so approximately 50% of dry mass are actually proteins. Proteins are a rather heterogeneous group of molecules exhibiting various functions. At this point, I usually ask my students when we have live sessions to name any examples of proteins they can think of and to try to define their biological function. And I can do that right now. So you can pause the video and think a little bit about it, whether you can think of any proteins and their functions. And I'm going to give you a couple of examples. Probably you have all heard about hemoglobin. It is an important molecule which has and exhibits transport role, transporting gases throughout our system and blood. Then probably you have heard about collagen and keratins, forming our hairs and nails and connective tissues and organs and everything. So they have a structural role. Probably you have heard about actin and myosin involved in the contraction, or enzymes catalyzing chemical reactions in our body, and so on and so on. So you see that there are numerous functions, and you can assume that probably the structure of these proteins is pretty diverse. Also, we can say that proteins are instruments of genetic information expression, and I'm going to explain this statement by the figure which is presented on the slide. So you all know that actually genes in our DNA contain codes for amino acids, or triplets of nitrogen bases or nucleotides, which are called codes, and they're going to define which amino acids are going to be incorporated in the molecule of protein. This is achieved through two processes. The first one is transcription, where the sequence of nucleotides is transcribed or transferred to the sequence of nucleotides on messenger RNA. Messenger RNA containing codons for amino acid sequences, where each codon or triplet of nucleotides defines a specific amino acid which is going to be incorporated into the structure of a future protein in the process of proteosynthesis of translation. So if you can see this pattern or this transfer, so information from genes are directly transferred to the sequence of amino acids in proteins. So proteins are those instruments which are used to express all the information encoded or incorporated in the DNA. And then synthesized proteins are going to coordinate and decide which other processes and molecules are going to be established or synthesized or whatever else. But the common characteristic for each and every peptide and protein is that they are polymers of those 20 proteinogenic amino acids that we have analyzed in the previous video. So when amino acids are polymerized, first we have the formation of the smaller units called peptides, and upon the further polymerization we get larger molecules which are marked as proteins. So now the question is, where is the limit? So where is and what is the point after which peptides becomes proteins. So the point is set at 10 to 5 daltons of molecular weight. So molecules with smaller molecular mass are defined as peptides, and larger molecules are defined as proteins. Why 10 to 5 daltons? This molecular weight and this number is actually picked up because of the fact that smaller molecules than 10 to 5, they can freely diffuse through membrane during dialysis, and larger molecules cannot do that. But what is the common characteristic, despite the fact whether we are talking about peptides or polypeptides or proteins, is that amino acids are attached to each other by the formation of one very specific bond, which is marked as peptide bond. Looking at the formation of a peptide bond, we can say that it's the reaction of polymerization, because a large number of amino acids are connected to form more complex structures. It's the reaction of dehydration, because during the formation of a peptide bond, and you will see it in a couple of seconds on the slide, one molecule of water is formed and released from the system, or removed from the system. And it's the reaction during which amide bond is formed. And if you know, and if you remember your organic chemistry course, you mentioned that amide bond is formed upon reaction of carboxylic and amino group. And this is what actually goes on in the formation of the peptide bond. So peptide bond is formed upon the reaction of alpha carboxylic group of one amino acid and alpha amino group of another amino acid, and here is the reaction. So let's analyze it. So here we have the carboxyl group of one amino acid, and this is amino group of another amino acid. So OH group from carboxylic group will combine with one of these two hydrogens from amino group, thus forming water, which is going to be released from the system, and the rest of the molecule is going to be attached by a chemical bond which we called peptide bond. So this is green shaded part of the molecule, COONH. This is the structure of a peptide bond, and we're going to discuss a little bit about some general characteristics of peptide bonds and peptides. So here we have the same thing, just represented in a little bit different way. So OH group from COOH group and H from NH2 group combine to form water, and the rest of the molecule tend to form peptide bond, and you can continue the sequence, thus adding the third amino acid, reacting in the same way, OH from the COOH and H from the NH2, forming one more second peptide bond, thus forming larger molecule, and you can continue the sequence. So when two amino acids are actually attached to each other, we have the formation of so-called dipeptides. And at this very moment, we can say some general rules regarding peptides and regarding their namings. So how can we form the name of a peptide? We are dealing with specific suffixes, and that specific suffix is YL, which is added at the end of the name of an amino acid in a way that each amino acid in a sequence, in a peptide, changes its suffix INE to YL, as you can see here, and only the last amino acid keeps its original name, so there is no change with the last amino acid. And I'm going to give you one example. I hope it's illustrative. So glycine and alanine react to form peptide bond. So the future dipeptide to be formed is going to have the name where glycine loses its suffix INE and is changed to YL, but alanine, as the last amino acid in a sequence, will remain unchanged. So the name of the dipeptide will be glycylalanine. If we continue the process by adding the third amino acid, for example methionine, then alanine will not be the last amino acid anymore, so we're gonna change its suffix I and E again to YL, and methionine as the last one remains unchanged. So the name of the future molecule and peptide will be glycyl alanine methionine. You can continue the sequence and make your own examples. If you tend to form and polymerize 3 amino acids in a sequence, then we get tripeptides. If you combine 4 amino acids, they tend to form tetrapeptides. And we can continue with the sequence: pentapeptides, hexapeptides, hectapeptides, octapeptides, etc., etc. We can also introduce here one new term, which is term oligopeptide. That is a peptide containing from 2 to 10 amino acids. And by further polymerization, we get polypeptides, meaning more amino acids are attached with more peptide bonds formed, and finally huge molecules, proteins. Notice here that the number of peptide bonds in certain peptide is always -1 to the number of amino acids. So dipeptide has 1 peptide bond, tripeptide has 2 peptide bonds, and so on and so on. Continue the sequence and make your own examples. Let's name and let's list some other rules. This one is easy. Amino acids are listed from the first to the last, from left to the right. So I have provided you here with one example of 1, 2, 3, 4, 5. It's pentapeptide. So let's take a look at this. 1, 2, 3, 4 peptide bonds. So pentapeptide, 4 peptide bonds. So amino acids are listed from the first to the last, from left to right. So in this sequence, first amino acid is, if you remember the structure, this is serine, then this is glycine, this is tyrosine, alanine, and so on. So you notice that in each peptide or polypeptide chain, we have 2 domains. The first domain is linear part of the molecule, and the second domain consists of those side chain groups, hydrogens and carbonyl groups, which are positioned above or below the linear part of the molecule. The linear part of the molecule contains one repeating sequence, which is C-alpha, alpha carbon atom carrying the side chain, C-alpha, C, N, C-alpha, C, C, N, C, C, N, C, C, N, and so on. So this repeating sequence C, C, N, C is marked as polypeptide backbone, and the rest of the groups are positioned above or below this polypeptide backbone. The other thing which can be noticed for each polypeptide is that the first amino acid in the sequence always has free amino group. And this part of the molecule is marked as N-end, N-terminus, and the first amino acid in the sequence is marked as N-terminal amino acid. The last amino acid always has free carboxyl group, so it's marked analogous to the previous one, like C-end, C-terminus, and this last amino acid is C-terminal amino acid. Let's move on. Let's analyze the peptide bond. I have extracted here the peptide bond itself. I don't have to distract your attention with the rest of the structure, which is at this moment is not that important. So usually we mark peptide bond as a single bond, but if we analyze the components of this peptide bond a little bit more detailed, we can notice some other possibilities. You all know from your chemistry course that nitrogen belongs to the 5th periodic group of elements. So it has 5 valent electrons, from which 3 are unpaired, and you have 1 free electron pair. And this electron pair here can be involved and introduced in the formation of 1 more coordinative covalent bond between nitrogen and carbon atom, which is gonna cause automatically the move of these electrons from carbonyl group towards electronegative oxygen, thus making the following situation. Nitrogen becomes positively charged because of the lack of those electron pair, which is involved in the formation of a double bond with carbon atom, and oxygen becomes negatively charged because of the move of the carbonyl electrons towards the oxygen. So now the question is, which of these 2 forms is correct one? A single bond or double bond? So the question is peptide bond single bond or double bond? And usually when we have live sessions, we have a discussion regarding this. And usually half of the group votes for a single bond and the other half of the group votes for a double bond. But actually both groups are right and both answers are correct. So peptide bond can be considered either a single bond or a double bond, or it can be considered both single and double, meaning that the truth is somewhere in between. And we say, chemically speaking, that peptide bond has a partial double bond character, or more commonly we say it's 1.5 bond, which means that it's neither single nor double, but it's some kind of mixed, a little bit of single, a little bit of double. It has a character of a double, but it looks like single, and so on and so on. So some of the very important properties of peptide bond arise from this fact that it's 1.5 bond and that it has a character of a double bond itself. And I'm going to try to explain this. So, and again, I'm going to need your chemistry knowledge. So if you have a single bond, you have sp3 hybridization. And you know that when you have a single bond, I'm going to try to represent it. So atomic groups, and you have 3 more here, which are attached, they can rotate freely around this single bond. But if you have a double bond, your attached groups, 2 groups in this case, cannot move, cannot rotate. So what is the situation with our peptide bond? The situation is that due to the 1.5 bond character, there is no free rotation of the groups around the bond, which makes the future polypeptide chains a pretty rigid and constant and stable structure, so it cannot change and rotate freely in any way, which is very important for the stability and structure and function of proteins. Let's move on. Again, let's get back to your knowledge about single and double bonds. When atoms tend to form single bonds, they undergo certain types of hybridization. And for single bonds, we have a specific sp3 type of hybridization. So we have 4 sp3 orbitals, which are equivalent. And now the question is, what is the space orientation of those 4 orbitals? They contain electrons, so they have to be positioned in a way that they are as far as possible from each other to minimize the electron repulsion. So people figure it out that the best structure and orientation for 4 sp3 orbitals is at the angles of tetrahedral, so the structure is tetrahedral. But when you have a double bond, then you have sp2 hybridization type and 3 sp2 orbitals containing electrons. So when you have 3 orbitals, the best way to position these orbitals is towards the angles of a triangle. So the angle is 120 degrees. So you can put the whole structure in one plane. So having that in mind, we can conclude that peptide bond is planar due to the partial double bond character and not tetrahedral like assumed from the single bond character. The next one also arises, and it's represented here in the figure, also arises from the fact that it has a double bond character. So when we are talking about compounds containing double bond, we have one very specific type of isomerism, which is geometrical isomerism, with alpha carbon atoms oriented on the opposite sides or at the same sides. So we're talking about trans and cis configurations. When those alpha carbon atoms carrying side chains are on opposite sides, we have trans configuration. If they're on the same side, we have cis configuration. The question is now, which of these two forms is energetically more favorable? And obviously, always we have a discussion in a live session, but this time I'm gonna do it myself, so it's easier and it's simpler and energetically more favorable if you have those huge group on opposite sides, because if they're at the same side, probably they're gonna collide and they're gonna bother each other, and it's gonna you're gonna have a lot of negative energy between them, which destabilize the system. But if they are positioned every and each of those on their own sides, it's energetically more favorable and you have the peace in the house. So trans configuration actually predominates in the structure of peptide bond. Cis configuration can be found only if some small amino acids are involved in the formation of such a bond, like glycine or alanine or something smaller. And the last property of the peptide bond, which arises from its structure, is its reactivity. So peptide bond is highly non-reactive, it's very stable and resistant to the action of numerous reagents. So if you want to break peptide bond, you have to introduce high temperature above 100 degrees centigrade. You have to expose it to the action of strong acids like sulfuric acid, and you have to cook it and boil it and do it for at least 24 hours, and then you can hydrolyze or break peptide bond, which is very good, which gives our proteins huge stability, and it keeps our integrity. Otherwise, we would easily react with the environment and with the systems in our environment, which would be not good at all. But our organism, on the other side, has a very filigrane, very nice mechanisms to smoothly dissociate or hydrolyze the peptide bond, and those are specific enzymes. But and of course you don't need any kind of extreme conditions to break down peptide bond by enzymes, but we're gonna discuss about those specific enzymes and peptide bond hydrolysis when the time comes in some of the latter chapters. Anyway, this is the summary of the facts that I have told you so, and this is actually the proof that the previous statements are actually correct. And people calculated the length of the specific bonds. So the common length of a single bond is 153 angstroms, and the common length of double bond is 124 angstroms. And when people calculated the length and determined the length of the peptide bond, it was at the point 1.32, which is exactly in the middle between 124 and 153, which actually proves that peptide bond is somewhere in between the single and the double bond. And this is the summary. So we have a sequence CCNC, we have a peptide bond, which is 1.5 bond, and electrons evenly distributed throughout the bond. It's planar. The rest of the groups are above or below the linear part of the molecule, and so on and so on. This slide actually represents some of the examples of biologically active peptides. I'm not going to go into details because by accident or accidentally. Most of these examples are examples of different peptide hormones which are involved in the regulation of numerous processes in our body. So we will have time later on to discuss about each and every of these hormones. So you can just read it through as an illustration, and I'm not going to go into details. Maybe I can put some attention to the first molecule, which is not a hormone, it's a glutathione 1,3 peptide containing glutamic acid, cysteine, and glycine, and its chemical name is gamma-glutamyl cysteinyl glycine. And actually the active group of this molecule is SH group, thiol group from the cysteine. And glutathione is found in all cells in human organism, and it's involved in the maintenance of the metabolism of SH group, or in other more acceptable words, we can say that glutathione is one of the most potent endogenous antioxidant systems which each cell has. So it due to its redox properties and the phenomenon that SH group can oxidize to form disulfide bond, glutathione can prevent the harmful effects of reactive oxygen species and free radicals, thus preventing the development of oxidative stress and, of course, subsequent disintegration of cells and cell membranes and organelles, and damages of biomolecules and so on, which might cause oxidative stress, and which at the end point leads to development of pathological processes and diseases. So yeah, we're gonna have it maybe at 1 or 2 points in the future, but I think that that glutathione is valuable mentioning here. So what do we know so far? So far we only know that amino acids are connected by peptide bonds, but we don't know anything else about the structure of proteins. And obviously knowing that there is a peptide bond involved is not enough, because knowing only this protein molecule actually tends to look like a boiled spaghetti, so it's not consistent. It tends to be reshaped in different ways, so obviously it doesn't have any consistency, and our body and our proteins and our structure are pretty stable, pretty constant, pretty consistent. So there has to be something else. There has to be some upgrade of the system, and that is the truth. So there are 4 levels of protein structural organization marked by the terms primary, secondary, tertiary, and quaternary structure. Secondary and tertiary structure actually they tend to affect the final structure and the biological function of the protein molecule the most. So usually we use one term, which is native conformation, which actually describes the shape and the structure of the molecule, and followed by its biological function. So this is a new term for us. So let's start from the beginning. Primary structure actually defines the amino acid sequence in a protein, and this is what we did so far. Those are amino acids connected by peptide bonds. So the amino acid sequence actually means knowing which amino acid is in the position number 1, which is in the position number 2, 3, 5, 28, 372, and so on. Knowing the primary structure is very important due to several reasons. I mentioned at the very beginning that the sequence of amino acid is directly connected to the sequence of nucleotides on DNA. So if a mutation happens, the consequence of that mutation can be the change in the structure of amino acid in a specific protein. And those changes, those changes can be various. So you can have a change where one amino acid is going to be changed to amino acid which is very similar to the previous one, and those are going to be called silent mutations, because as a consequence of this change, you won't have a huge change in the structure or the function of a molecule, which is very important for evolution. On the other side, you can have the change in amino acids which are dramatic. For example, if hydrophilic amino acid is changed to hydrophobic, the one liking water, the other one tending to get away from the molecule, so water, thus absolutely affecting the structure and subsequently the function of the protein molecule. And I'm going to give you one example of this. There is one molecule, and you will guess which molecule is that. And due to the mutation on DNA, the situation happens that one amino acid at position 6 on just one type of the polypeptide chain out of 2 types and 4 chains involved in the structure. So only one amino acid at the position number 6, and that is glutamic acid, is changed for valine, and which means that hydrophilic acid, liking water and orienting towards the aqueous medium, is changed for hydrophobic amino acid, which doesn't like water and tries to get away from it. So one amino acid which was at the surface now tends to get away from the water to the central part of the molecule, thus absolutely significantly changing the structure of the overall molecule from one globular form to something more like discoidal form. And obviously it's going to affect the function as well, and it's going to mean the absolute loss of the function of this molecule. So can you guess which molecule I'm talking about? Yeah, it's hemoglobin, and it's sickle cell anemia. So only the change in one amino acid may cause dramatic changes in the structure and in the function of the molecule, and in the end you're going to get an absolutely dysfunctional molecule, unable to transport gases and to bind oxygen and carbon dioxide and so on. Primary structure is also important from the evolution point. So species which are far away from each other on an evolution scale, they're going to have rather different proteins, and species which are close to each other on an evolution scale, they're going to have rather similar proteins. And actually that was the trigger for some scientists to try to analyze amino acid sequences in certain molecules of the same function in different species which are believed to be close on an evolution scale. So Sanger analyzed the amino acid sequence in insulin in human organism and in pigs, and for that he actually won the Nobel Prize as well in 1953 because he actually concluded that the amino acid sequences of pigs and human insulin differ in only a few slight changes which are not important, which are not crucial, not affecting the structure and the function of the insulin. So it was a revolution in treatment of diabetes mellitus, really worth winning the Nobel Prize. And because of that importance, I'm going to tell you just in brief how the amino acid sequence can be determined in one protein. So there are a couple of stages or a couple of steps in doing such a process, and it's time-consuming and complicated process. So the first, you have to start with the cleavage of polypeptide to smaller fragments. So if you have a huge polymer complex protein molecule containing of more than one polypeptide chain, first you have to separate those polypeptide chains. Then, in order to successfully manipulate with the material, you cannot manipulate with a chain containing several hundreds or thousands of molecules. So you have to break it into smaller fragments, or you have to cleave it into smaller fragments. And for that purpose, you can use enzymes. for example, trypsin, chymotrypsin, which are very specific, so they tend to break and cleave specific peptide bonds between specific amino acids. So you can follow your process when you have to put the puzzle back together. And then you have to mark those fragments with cyanogen bromide, but I'm not going to torture you with the details. And then when you identify those fragments, you have to play like a puzzle. You have to compare those fragments and try to find the overlapping sections of those fragments, thus enabling you to put them in a sequence. Okay, this fragment was first, this fragment was the second, this one was the third, and so on. The second stage of this determination is to determine the amino acid content in proteins. So first you have to break peptide bonds, subjecting them to acidic hydrolysis to get all amino acids separated from each other. And then you perform one analytical technique, which is called chromatography, to separate different types of amino acids from each other and to have them in, let's say, separate test tubes. So medical students, we're going to discuss about chromatography in details on our practicals. Dentistry students, we're gonna skip that because it's too much for your course. So you just have to, for now, remember that chromatography can be used to separate and to isolate different types of amino acids from each other. So you have them like alanine here, glycine here, phenylalanine here, and so on. And then you have to quantify those amino acids to see what is the concentration or how many molecules of this amino acid you have or that amino acid you have in that specific fragment. And you can achieve it with ninhydrin. With both groups, we're going to talk about the quantification of peptides and proteins at the practicals, and I'm going to elaborate this in more details at the practicals, but for now just with ninhydrin. The third reaction, the third step, the third stage is to determine N-terminal amino acids, so the first one in the sequence. And you can do it by Sanger reagent, which is fluorodinitrobenzene, which works in a way that the reagent is attached to the N-terminal amino acid and the rest of the amino acids are hydrolyzed or dissociated, and this first N-terminal amino acid attached to the reagent, it has a very specific chromatographical properties, and it differs from the rest of the amino acids, so it can be isolated and defined very easily. The second version is Edman reagent. It's even better than the Sanger because phenyl isothiocyanate is attached again to the N-terminal amino acid and removes it from the fragment, but the rest of the fragment remains untouched, so you can add the new aliquot, the new portion of the reagent, which is gonna remove the next N-terminal amino acid, and so on and so on, so you can precisely determine which was the first, the second, the third, and so on. You can do it from the other way, from the opposite side of the polypeptide chain, so you can subject your fragment to the determination of C-terminal amino acid. You can use hydrazine, which actually reacts with all amino acid but the C-terminal, and you can use an enzyme, carboxypeptidase, which is gonna remove C-terminal amino acid from the rest of the fragment, and you can separate it by chromatography and identify it. Anyway, but still we don't know anything more about the structure, so that polypeptide chain with the specific amino acid sequence has to be upgraded, has to be organized in a different way, and the next level of organization is secondary protein structure. We have two options, alpha helix and beta pleated sheet. We have some more additional options, alternative options like triple helix in collagen fibers, but for now I'm not going to discuss about those. Medical students are going to do it when doing collagen and keratin and connective tissue biochemistry, and dentistry students, you're not going to need it for your level, course level. So let's start with alpha helix. So you have it here. So polypeptide chain tend to form a helix, and approximately 3.6 amino acids are found in one coil of the helix. Stabilization of the structure is achieved by the formation of hydrogen bonds. So despite the fact that those hydrogen bonds are weak interactions, because of the fact that they are formed between different peptide bonds, and you have hundreds and thousands of peptide bonds in large proteins, so a huge number of those hydrogen bonds actually results in a significant stabilization potential. So one hydrogen bond itself is rather weak interaction, represents insignificant stabilization tool, but if you have a lot of them, then the stabilization potential really increases. We have some exceptions. Proline amino acid, proline, our amino acid, does not support alpha helix. Also, it is believed and it's suggested that voluminous amino acids with voluminous side chains are kind of incompatible. They're not impossible to be found in alpha helix, but they're incompatible with it, like glutamic acid, aspartic acid, those basic amino acids, histidine, lysine, arginine, if you remember, they're huge side chains, or valine, leucine, tryptophan, and so on. Actually, in any way, whether they like or don't like the alpha helical distribution, side chain groups are usually oriented towards the surface of the alpha helix. So if you cannot form alpha helix, you can certainly form beta pleated sheet. And in this level type of organization, secondary organization, the chains are organized in the form of an adjacent parallel strands, so they tend to form sheets. So in order to prevent sliding of the sheets over one another, the structures are also stabilized by the formation of numerous hydrogen bonds between the peptide bonds. And now we can go into details. So there are a lot of subgroups of beta pleated sheet organization. I have mentioned only a couple of those. So it can be formed between two different polypeptide chains, or it can be formed inside a single strand. They can be parallel, or they can be anti-parallel. They can tend to form some super foiled structures and so on and so on. But you really, at this level of the course, you don't have to go into so many details. Maybe you will grab some of these information later on when evaluating some specific proteins, but really for now, don't bother yourself with going into details about the different subversions of the beta pleated sheet structures. Tertiary structure is, again, the next level, upper level, upgraded level of protein structure, and actually it describes the final strand shape in space, because knowing that you have an alpha helix or beta pleated sheet, still you can fold that structure in huge in various ways. So the final 3D structure, three-dimensional structure, the space orientation of that strand in space actually is described by tertiary protein structure. And according to the general shapes of protein molecules, they can be classified into globular and fibrillar proteins, depending on whether they tend to form globular structures or fibrillar structures. And of course, like with the previous type of protein structure, we have to stabilize this structure and to prevent unfolding that shape of the molecule. And this is achieved by the formation of secondary bond types, and 4 of them are found in tertiary protein structure. Those are hydrophobic interactions, which I have already explained previously, which actually means that hydrophobic amino acids, they tend to orient in the central part of the molecule, and hydrophilic amino acid, they tend to form they tend to orient towards the surface of the molecule, towards the aqueous medium. Hydrogen bonds, we have already explained it thousands of times. Ionic interactions are clear between the charged groups in the side chains. And this one is very specific, disulfide bond, or disulfide bridge, which is formed between upon the reaction of SH groups of cysteines, which can be adjacent, but they can be far away from each other in the polypeptide chain, thus causing this very specific folding of the molecule. And notice that this is the only covalent bonding in the secondary types of bonding. All other stabilization bonds are of the type of interactions. Then we have quaternary protein structure, and I like to say that it's optional. So only complex proteins which contain more than one polypeptide chains actually have quaternary protein structure. Actually, it describes the relationship between two or more polypeptide chains which are found in the structure of those complex proteins. And again, the quaternary structure has to be stabilized by secondary bond types. And here we also have hydrophobic interactions, hydrogen bonds formation, and ionic interactions, but we don't have disulfide bridges. So disulfide bonds are specific only and exclusively for tertiary structure. And to summarize the story, here we have the primary structure, so just the sequence of different amino acids, and then they tend to form secondary types of organization in the form of alpha helix or beta pleated sheet. And at this point, it's worth mentioning that in one polypeptide chain, you will find both secondary types of organizations. So you will find a little bit of alpha helix and then a little bit followed by a little bit of beta pleated sheet and so on, like in this figure. So you have a little bit of alpha helix, then a little bit of beta pleated sheet, and so on and so on. So the final shape of the strand in a space is described by tertiary structure, and if a molecule contains two or more polypeptide chains, the relationship between them is described by quaternary structure. Speaking of physicochemical properties of proteins, we can name a few. First, proteins are charged molecules, so they have a certain electrical charge, plus or minus, depending on the nature of the groups in the side chain. So they are charged molecules. Due to a huge molecular mass, they tend to form colloid solutions, which you know are pretty unstable. So there is a question whether the size can affect or represent the solubility problem for proteins. And we might say yes, but on the other side, we have a lot of body fluids in which numerous proteins are nicely and pretty stable dissolved in that aqueous medium. So we have to explain the mechanism which enables the solubility of proteins, nice and decent solubility of proteins in water. So what is the point? The point is the same as with dissolution of any other molecule. For example, when you studied chemistry, you started the dissolution with the most simple example of sodium chloride in water. So sodium and chloride ions are interact with the molecules of water, and dipoles of water surrounds these ions, forming hydration layer. So the same thing happens with proteins. So having in mind that they are charged molecules, so they are going to surround themselves by the dipoles of water, or hydration layer or hydration shell is going to be formed, like represented here in a figure. And this hydration layer is going to be some kind of protective zone which has to prevent the ability of these proteins to interconnect, to to associate in huge aggregates and to precipitate and further on destabilize the system. So isoelectric point is also one of the properties which is transferred from amino acids. So it's a pH at which protein molecule becomes zero charged, it becomes neutral, so its solubility decreases significantly and it cannot move in electrical field anymore. So exactly the same like we had with amino acid. We introduced the phenomenon of native conformation, but now we can introduce one more term, and that is denaturation, which means that protein molecule can be affected in a way that it loses its native conformation, which means that it loses its structure and subsequently its biological function. Sometimes proteins can be renaturated or they can be brought back to the initial state, but in most cases they cannot. So, speaking of physico-chemical properties of proteins, one more fact is important and worth mentioning here, and that is the isolation or precipitation of proteins, which can be irreversible in cases when we're going to have a denaturation of proteins. Without any possibilities to bring the protein molecule back to the initial state, so it's destroyed for good. And we have a reversible precipitation of proteins, after which we can bring back both the structure and biological function to the protein, so they're gonna remain untouched. So speaking of irreversible ways to denaturate or precipitate proteins, I have illustrated a few. So exposure to strong acids and strong bases causes denaturation of proteins, but during that exposure peptide bonds are not affected, but rather quaternary, tertiary and secondary structure are destroyed, thus losing the biological function. Exposure to high temperature also affects the protein structure and function. The increase of the temperature actually increases the solubility of a protein in an aqueous solution up to kind of 40 degrees, but after 40 degrees solubility drops down, and after 50 degrees denaturation occurs and a protein loses its native conformation. Addition of heavy metal salts to the protein solution, heavy metals like lead, like mercury, like cadmium, so they all tend to form very insoluble disulfides, and you know that we have disulfides bridges. So after exposure to heavy metals, proteins are denaturated for good, permanently denaturated, and you cannot bring them back to the initial state. And actually this fact actually lies in the basis of the toxicity of those heavy metals. Organic solvents also may cause denaturation of proteins. For example, ethanol actually causes denaturation, which is one more additional point to its toxicity. And mechanical effects, when you expose the protein solution to some mechanical action, for example, like preparing the egg yolk schnee or egg white schnee, you cannot get it back if you make a mistake. So mechanical effects permanently destroys the structure and the function of a protein. But from the point of view of laboratory stuff and issues, sometimes it is very valuable to isolate protein or precipitate protein from the solution, but in a way to prevent the loss of its structure and biological activity. So to make the precipitation reversible, which enables you to bring the protein back to its initial state after isolation, after precipitation, thus not affecting its native conformation. And this can be achieved in two ways. The first method is salting out, and the second one is the achievement of isoelectric point, which I have previously described. What is the salting out technique? Actually, its name says the precipitation is a result of the addition of neutral salts in a high concentration. So when you add one salt, which usually contains either sodium, potassium, or ammonium ions, or cations, or chloride, sulfate, phosphate, anions. So if you add them in a high concentration, they tend to form hydration layer themselves, like here. So they tend to surround themselves with deep pools of water as well, like proteins. But if you add a huge concentration of those, and ionic strength of those small ions is much higher than the strength of the protein, which these charges are widespread on a huge volume in a huge space. So actually the competition starts, and salt ions are winning. They're actually taking away deep pools of water from the protein hydration shell, thus leaving them, let's say, kind of naked. So protein molecules which are charged cannot be attracted to each other, forming huge aggregates, and which can lead to the precipitation due to the overweight. But notice that we targeted only the hydration shell, the hydration layer. We haven't affected the structure and, of course, possibly and probably the biological function of the protein. So when you add a little bit of water, or when you add a diluted salt solution, you can bring back and you can recover the protein and bring it back to its initial state, and it will probably keep its biological function as well. In order to make this salting out process quantitative and more effective, you can combine salting out with the achievement of isoelectric point, which we said leads to the decrease in the solubility of the protein itself. Speaking of classification of proteins, there are different ways to classify proteins. The first one is according to the tertiary structure, and I have already mentioned this. And according to this, proteins are classified to globular proteins. They tend to form globules or balls. And fibrillar proteins or fibrous proteins, they tend to form fibrils. The second way to classify is according to the content of a protein. And according to this, proteins are classified to homoproteins. Those are proteins containing only amino acids in their structure. And heteroproteins, those are more complex proteins, more heterogeneous proteins. So apart from the amino acid part of the molecule, which is also marked as apoprotein, they tend to have some other molecules attached to it, and those are marked as prosthetic groups. Speaking of homoproteins, we have 6 classes of homoproteins: albumins, globulins, prolamins, protamins, histones, and scleroproteins. I'm not going to bother you with the rest of the types, but I would like to mention albumin and globulins, because those are the major, at the same time, they are the major fractions of our blood plasma proteins. So albumins are soluble in water, and they can be precipitate upon the addition of saturated salt solutions. On the other side, globulins are insoluble in pure water, but they are soluble in diluted salt solutions, and they precipitate upon the addition of half-saturated salt solutions, and these properties are used in a lab for separation of these fractions, and we're going to need these facts later on for practicals. Speaking of heteroproteins, we also have various groups of those depending on the type of prosthetic group attached to the amino acid part. So we have heme proteins, where heme is a prosthetic group, and we have hemoglobin, myoglobin, cytochromes belonging to this group, and you are going to have separate lectures about those. Metalloproteins, where you have metal ion in the structure, so you're going to do transferrin, ferritin, and so on. Glycoproteins, they have some sugar components attached to the protein part of the molecule, which can be mono or oligosaccharides. phosphoproteins with the phosphate group as a prosthetic group, lipoproteins with lipid molecules as prosthetic groups, and you're going to have a separate lecture about glycoproteins, phosphoproteins, and lipoproteins as well later on. Nucleoproteins with nucleic acid as a prosthetic group, and we're going to discuss about DNA and RNA later on. The third and the final way, and maybe the most common and most favorable nowadays, is according to the function of the protein. And according to this, proteins are classified in 10 groups. So we have already mentioned some of them. Enzymes as biocatalysts, transport proteins, we mentioned hemoglobin, we mentioned transferrin, we mentioned some of the proteins specified for transport of certain molecules. There are a lot of them. Contractile proteins, I think that we have mentioned actin, myosin. Structure proteins, we mentioned collagen, keratin, elastin, and so on. Hormones, we have a whole table full of hormones, and we're gonna discuss about these a lot throughout this biochem course. Then receptors, they are proteins, and they enable the cell to communicate with other cells or to communicate with the environment, to accept and receive the information, to send the information and signals and so on from other molecules and so on. Antibodies, which are very famous nowadays during this COVID-19 pandemics, and we're going to do just a little bit about it, about the immune response, about the immunoglobulins and so on. Then growth factors and gene regulatory proteins with very specific functions, and toxins as the last group, because a lot of toxins which can be either synthesized, endogenously synthesized in our body during metabolic pathways, or exogenously taken as xenobiotics from the environment, they tend to be of protein or peptide structure. Some of them, not all of them, but some of them. For example, like bee poison, snake poison, mushroom poison, and some of the others are actually of a peptide origin. Okay, anyway, girls and guys, my dear audience, that was more or less it about proteins. I know it was a lot, but the chapter is huge, so I think it was acceptably interesting. I think it was useful, and I hope it won't be a big deal for you in memorizing all these facts and nice stories, which are actually the preparation for proper understanding of the upcoming more complex chapters. So far I'm gonna say goodbye, and until seeing you in some other video clips, bye bye, have a nice rest of the day.